Hey, this is Mrs. Dellosso, and in this podcast, we're going to continue talking about organic compounds. We will specifically focus on proteins and nucleic acids, and I grouped these two organic compounds together because proteins and nucleic acids are actually codependent. And we'll talk a little bit more about that in a moment. We just finished talking about carbohydrates and lipids in the last podcast, and carbohydrates and lipids are pretty much going to be the same, even in different organisms. But proteins, although similar in structure and function, are different. Although in some organisms, some proteins will be the same. For example, don't mind this whole big picture here, but I just wanted to pinpoint to you or show you insulin. Insulin is a protein that metabolizes food. And if we were to take the insulin protein from humans, we would see that it's pretty much identical to the protein insulin found in animals. However, though, human hair protein is different than the protein found in animal hair. And even so, even among people, one person's hair proteins may be similar to another's, but they won't be identical. So the uniqueness of an organism's proteins is the result of nucleic acids, which are found here, or RNA and DNA, and the enzymes. So nucleic acids contain the code to manufacture proteins. Then enzymes, which are proteins, help the nucleic acids function properly. And this is why proteins and nucleic acids are codependent. So let's break it down. Let's figure out what proteins are made up of, and let's take a deeper look into their structure and function. So proteins mainly consist of the elements carbon, hydrogen, oxygen, and nitrogen. Sometimes they may include phosphorus, sulfur, and a few other elements, but their monomers or building blocks are amino acids. So when we have amino acids joined together, they'll form a protein. So the function of proteins are, some will function as enzymes, which we know catalyze reactions, or they can function as structural building blocks, giving structures to cells, for example. So that's just an overview of proteins, what they're made up of, and their main job or role. Let's take a deeper look into an amino acid. So here's a diagram representing amino acid structure, and there's going to be, well, kind of 4 different parts here. So in an amino acid, we're always going to find an amino group, a carboxyl group, the carbon backbone, and a side chain or R group, which will make each amino acid different. So depending on the side chain or the R group here will depend on what amino acid there is. And there are 20 different amino acids that we can find, and which means there can be so many different possibilities and arrangements of amino acids to form many, many different proteins. How are amino acids joined together? So on the left here we have one amino acid, and to link it to another amino acid will be the process of dehydration synthesis. Now recall, dehydration synthesis means that we're going to remove a water molecule in order to combine 2 molecules together. So amino acids will join together by the process of dehydration synthesis. Here's another diagram showing how amino acids will be joined together by the removal of a water molecule, which is the process of dehydration synthesis, and then the bond that's going to form in between the amino acids is called a peptide bond. So here we have 2 amino acids, which would be a dipeptide bond, but if we were to continue to add amino acids and link them together, we would then form something called a polypeptide chain. And eventually, this polypeptide chain will ultimately come to look like this 3-dimensional shape here. So how does that happen? So we have the polypeptide chain over here on the left, which joins the amino acids together by the process of dehydration synthesis. Then eventually, diagram 2 here, the polypeptide chain is going to coil or even fold in a sheet-like arrangement. Then third here, the coiled or folded structure itself is then folded again and held in a precise shape. And then last, the peptide chains may lock together to form a protein. And now the polypeptide chain has a three-dimensional arrangement, and it is this shape that forms the active sites of the enzymatic proteins. So having the active site, now substrates can then bind with the enzyme during a chemical reaction. But if some of the amino acids are not in the right sequence, that could alter the shape of the protein, which could have an effect on the active site. It might change the shape and the ability of a substrate to bind to that. So the proper arrangement of the protein and the amino acid sequence is very, very key when it comes to forming the protein, because if the shape is altered, then that will not allow the protein to carry out its job. All right, now we're going to take a look at nucleic acids. Right from the beginning, I told you the two examples of nucleic acids are RNA and DNA is found within the nucleus of the cell. And it wasn't until just before World War I that they discovered that nucleic acids, not proteins, are the hereditary material. In other words, it's the DNA that transmits the characteristics to another cell. So a cell must pass nucleic acids to new cells in order for them to know how to function. So nucleic acids, DNA, and RNA are found in the nucleus, and they direct the activities of a cell by guiding the formation of both structural and enzymatic proteins. So nucleic acids, in other words, contain the code in order to make proteins. So by the time of World War II, scientists analyzed the components of DNA, but were really confused on the exact arrangement of the components of DNA. So really, to understand how DNA functions, it was really important to develop a model of its components. And in 1953, Watson and Crick, the two scientists of the left, came up with the model of DNA, which is known as the double helix. So they used X-ray information from DNA to help them come up with this model. And the scientist on the right, Rosalind Franklin, also obtained X-ray information from DNA, which really gave helpful information to Watson and Crick to help develop this model. And this model, the double helix, is still used today, although slightly modified from the original version. All right, so now we're going to focus specifically on the nucleic acid DNA. So what are the building blocks or monomers of DNA? DNA contains a phosphate, which is a small molecule made of phosphorus and oxygen atoms. It contains a 5-carbon sugar called deoxyribose, which gives it its name. DNA stands for deoxyribonucleic acid. Deoxyribose. And it contains a base. So each nucleotide will have one of the 4 bases, A, T, C, or G. So if this represents one nucleotide here, this base could be A. Now on the other side, remember DNA is a double helix, so there's 2 strands. So this would pair up with another nucleotide, and if you remember, following the base rules, A will always pair with T and C will always pair with G. So what's the function of DNA? Well, DNA really controls all of cells' activities, but also contains the information in order to make proteins. So all of the cell's proteins come from the instructions in DNA, and this process we're going to talk about a little bit later on in the year. And what you're looking at in this diagram here is DNA replicating itself. So if you notice, there's enzymes that are involved in untwisting or unwinding the DNA. And over here you'll see the DNA bases being added here. And the result will be 2 identical DNA molecules. So on this side we'll have another set of DNA, and on this side another DNA. All right, now we're going to talk about RNA. So the monomers, or the building blocks of RNA, are pretty much the same as in DNA. But if you look at DNA here, we notice that it's only single-stranded, it's not double-stranded, and it still contains 4 nitrogenous bases, except instead of thymine, we're going to find uracil instead. Another difference is the sugar. DNA contains the sugar deoxyribose. The sugar in RNA is called ribose. So RNA stands for ribonucleic acid. So what's the function of RNA? From DNA, remember we talked about how DNA contains the instructions in order to make proteins. Well, from DNA we will actually form RNA, which will then contain the code to make the protein. So from DNA to protein, we must need the molecule of RNA. So RNA will then be used to make the protein. So how is the RNA actually formed? There is an enzyme called RNA polymerase that's going to start at a specific section of the DNA molecule, and then it's going to move down the DNA molecule, causing a section of the DNA to separate. So the 2 strands of DNA will then separate, and then other enzymes are going to pair the proper RNA molecules to the DNA. Then eventually the RNA chain will separate from the DNA, and then the DNA strand will then reform. So this RNA will then leave the nucleus and travel to a different site in the cell in order to help make the protein. Alright, so this concludes our video on the organic compounds, proteins, and nucleic acids. Hope you found this video helpful, and remember, always bring your questions with you to class.